All right. So first of all, I would like to highlight some of the key major challenges in terms of using antibodies against targets, where it's pedestrian in terms of generating a large number of antibodies against novel targets, and it's a very long process and a very costly process in terms of generating the antibodies against novel targets. So this is where it has high cost to generate these antibodies, and also because of the size of the antibody, they are around 150 kilodalton. So in terms of the penetration of the tissue, it's more challenging in terms of especially getting into intracellular targets. And for the antibody, so some of them, they are still in more polyclonal, so there is variability in terms of the quality of the antibody that you are using, and it requires the generation using animal to generate the antibody. And so there's challenges in the area where there are ethics challenges, where more of the movement into the animal-free research kind of program, and also antibodies that have Fc and isotype background, where the Fc and isotypes, they can cause non-specific staining, as well as they might be also having functions that could actually interfere with the signal of the assay. And also with the Fc of the antibody, that would also be affecting in terms of that you would need to have additional time in terms of doing the blocking step against the Fc before using it to stain onto the sample. And also the antibody, they are non-recombinant by default. So after identifying the clones of the sequence of the antibody, we will then need to then make them recombinantly. So this is where, with all these challenges in the biology space and in the context of the biotherapeutics, is that there is a lack of reagents, that there are more and more novel protein targets that we need to rapidly need more technology to better understand the biology of these proteins, and that allows us to better understand the mode of action, and in the context of the drug development, that can be very useful in the area of early target discovery. So this is where radically a new approach is needed. So our company, we have then developed this new technology, which is called the Ankyrons. So these Ankyrons, they are highly stable, which is just 15 kilodalton, and they are stable as a monomeric form, and they are made of ankyrin repeat scaffold protein. So how we select these binders, it's from our TeraLibrary, which contains of about 1 trillion clones in the library, and we use in vitro by ribosome display to select positive binders against the target of interest. So this is a very cost-effective and a high-throughput method to identify positive binders as a research tool against the protein that you are interested to work with, and the generation of multiple recombinant monoclonal binders with a defined sequence against a single target. So in terms of the application of the Ankyrons, pretty much they are very similar, or actually exactly the same as how you would use an antibody. So you can use in this range of different applications, and our team in the UK, we are working on putting lots of efforts in generating a protocol that works best for the Ankyrons. We can make suggestions on that, we can share our experience, and we are also doing a lot of effort in the area where we are validating multiple clones of the Ankyrons against a range of proteins that we have identified. So some of the advantage of the Ankyrons is that they are small, so the tissue penetration, especially for intracellular target, it's very effective. And in addition to that, being small, the proximity from the label to the binding site, it's much closer as compared to the antibody. And this has actually allowed us to actually generate a much higher resolution images when we are doing microscopy work, which I will share a little bit more in details of the data that we have generated. And typically in the clones that we have identified against binders, we are seeing that the binding affinity, that they are at picomolar to low nanomolar binding affinity, as you can see from the solution equilibrium titration that we perform against PD-L1 binders using the 3 clones of the Ankyrons, and you can see that they are all at the picomolar or the low nanomolar binding affinity. And using the Ankyrons, it allows you to save time, especially if you require to do a blocking step, because the Ankyrons, it doesn't have any Fc region, so there's no blocking step that is required when you are using it to run, especially in the ELISA method. So these Ankyrons, they are totally animal-free, so no immunization is required in the animal. So this helps to actually overcome a major constraint where if there were to be immunotolerant in the animal, there wouldn't be any immune response generated, meaning to say there wouldn't be any antibody against the protein of interest. So this is where the beauty of this technology that is fully recombinant. It does not involve any animal, so we are not constrained by any of this immunotolerant of using the immunization of the animal to generate antibody. And it doesn't have isotype and Fc domain, that's already mentioned. And these Ankyrons, they are all defined recombinant monoclonal sequences, and they are a highly consistent research reagent. And the good thing about the Ankyrons is that you do not have to think about switching all your other favorite antibodies that is already in your panel. This can actually seamlessly integrate with your current panel of antibodies in running your flow, your IHC, your IF. That can actually integrate very nicely with your panel of your cocktail of antibodies that you know that is really working well. So another key advantage of the Ankyrons is that it can distinguish, we can perform a guided selection to distinguish between very similar protein that just off by one amino acid, and that can increase the specificity of the clones that are generated against the protein of your interest. So that's where we can perform a guided selection process to identify clones that are more specific against the target of your interest instead of other protein that is closely related in terms of sequences. So, additional things about the benefit of the Ankyrons is that it's very flexible. So first of all, for the Ankyrons, when we first express them, it comes with a default V5 tag. So with the V5 tag, you then come in with an anti-V5 secondary. So you can decide on selecting the colors of the channels depending on the colors that you want in terms of your antibody cocktails, and we can also provide HRP where you can use it for IHC, so it's very flexible in terms of the labeling option. And afterwards, what we can actually make the Ankyrons is that we can make it into a direct labeling method where we don't have to think about secondary anymore. So what we do is that we will cleave the V5 and then we will then directly conjugate with the choice of the floral tag that you want to get onto the Ankyron. So you can directly stain onto your samples. So moving forward, there are many other applications that you can consider in terms using the Ankyrons, where you can actually link up Ankyrons of the same clone to increase the binding avidity against the target, or even we can actually make them bispecific, multispecific, and it's a plug and play technology. And for you to really understand what would be the downstream mechanism if you were to combine both specificity of the Ankyrons against two different targets. So it's a very flexible technology that we are able to offer in this aspect. So what I want to highlight here is that for the past 2 years, ProImmune, we have been putting a lot of resources and effort in terms of expanding our catalog range by screening against 10 over thousands of proteins, which we have now expanded our number of proteins in up to 3,500 proteins that we have screened, and the number of clones that we identified is more than 20,000 clones against these proteins across multiple species. So what we can do here is that I put a QR code of our catalog, which I take you through in terms of how you can actually search for these Ankyrons, these targets that you would like to have, and whether or not if they are on our catalog. And what we have done is that the Ankyrons that we screen against the target is that we are using an ELISA-based method. So we do a screening against the target, and from based on the signal to noise ratio, then we will then select those positive binders, and then those clones will then be put up onto the catalog. So this is how the catalog would look like if you go to our web page. So you can simply search by many different types of filtering. You can search basically by the entry name of your protein, or you can go by gene name, organism name, or the UniProt ID. That will then filter out if whether or not they are on our those targets is already on our catalog page. If not, we are welcoming in terms of doing customization. So we can actually do customization, and but one in order for this to happen is that we will of course need the antigen for us in order to do the screening. So this is where we set up a platform which is called the Ankyrons X, where we are able to help you to actually do the expression of the antigen, the protein target. So what we just need is basically we need to know what are the protein targets you are interested in. Let us know the UniProt ID. We can look into it. We can then express the protein. We will then do the screening against our Ankyrons library at the second step. And then afterwards, once we have identified positive binders, we can then sequence out and then we can then express them recombinantly as a research tool. So this right now, it takes us about 12 weeks to do the customization process, and we are trying to optimize this whole process and trying to reduce the overall turnaround time. So you can actually appreciate that if you have been in the area in terms of generating custom antibody, you can appreciate that how much time you are actually saving, as well as on the cost perspective when you are actually using generating the Ankyrons for custom novel targets, as well as even for the those that is already on our catalog. So I'm going to show you some data that we have generated using the Ankyrons that we generated internally and as well as our collaborators around the world. So this is data using immunofluorescence. So this is Ankyrons against low-density lipoprotein receptor-related protein 11, in short called the RRP11. So the challenge with this target is that there is no antibody against this protein. So that is where we have to think about alternative approaches and other alternative approaches and technologies that are able to actually address and able to target this and to really understand more about this protein. So that is where we then worked with this group and we generated the protein, we screened against the library, and we had the clones. And this is where you can see in terms of the data here. So this area here for the blue color, this is the DEP counter staining of the nuclei, and this is where you can see a very nice staining of the using the Ankyrons in red using using NTV5 APC. So this is another case study where we work with the Purbright Institute, and this is just showing, not just about, we want to think about, a wider variety of species of different targets. So this is where we work in understanding about the immune response of the mosquito. So this is specifically looking at the NF-kappa B. So I think there isn't, a lot of available tools, especially understanding about other species. So this is where we have Ankyrons that it doesn't discriminate any species, and we really want to work, with the tree of life of different species that we can actually better understand the mechanism. So this is where we generated the Ankyrons against the mosquito NF-kappa B. So you can see that from here, is that these are the wild type cells, and you can see that those white colors staining here, this is the Ankyrons staining onto the NF-kappa B. So they wanted to show that if whether or not is it specific to the NF-kappa B. So they perform a knockout experiment, and you can see that it's nice and clean that there isn't any NF-kappa B, and this was then published as a paper. So more case study in terms of other application in using flow cytometry. So this is again piezo1. So this is a Nobel-winning prize in physiology and also medicine in 2023. But there was a challenge that there wasn't any reliable existing antibody that could really look into the expression of these piezo1, which is a very important target to actually understand the roles between the ADAM17 and ADAM11 signaling. So this is where they use the Ankyrons to show in terms of doing the validation, where you could see that using the dark purple. So this is piezo1 rescue. So this is stained with the Ankyrons with the NTV5 secondary. You can see the signal here, and this is against the knockout of the piezo1. So you can see that the signal is at a lower range, and then this is just doing without the Ankyrons with just the NTV5 secondary, so you could not see any of the high signal. So this is another study that we performed with Purbright Institute again. So this is an area where there isn't any antibodies that's existing against the IgG3, even though there's antibodies against the pan-IgG of the bovine, for this is in cow. So they want to actually better understand about the role of IgG3. So they then gave us the challenge if we could actually identify clones that are only specific to IgG3 but not to IgG1 and IgG2. So this is where then we perform the screening, and you can see that from these ELISA data here. You can see this is two different clones. So you can see that for the gray color line here, this is representing the signal of the IgG3, and the blue and red, they are IgG1 and 2. So you can see that they are nice and flat at baseline. So you can see that doing a titratable effect using the Ankyrons to identify just the IgG3. It doesn't cross-react to the other IgGs. So this that also Ankyrons can be also used in the IHC. We have generated a lot of beautiful data, and just want to also highlight in terms of the Ankyrons, looking at the pancreas here. So targeting the insulin, so you can see a very nice islet here in terms of the staining of the using the Ankyrons. So in terms of Ankyrons, it helps to really accelerate the research. Just to summarize that they are small, they are just 15 kilodalton reagent. They are high binding affinity reagent against the target at picomolar to low nanomolar binding affinity. It's most importantly, it's very cost effective and also a very, very high throughput generation of the Ankyrons. And also, this allows you to actually target research areas that you want to understand a certain pathway of the biology of a protein that you are wanting to work with. So this is really a very cost-effective manner and a quick turnaround to actually get the tools that is required as you need to better understand the target that you are interested to work with. So right now, as I already mentioned, the catalog that we have over 20,000 of clones identified across 3,500 protein across multiple species of more than 150. So if you all have any targets that you are interested in, feel free to reach out to us, because I think what we really want to do is that we want to better understand what are some of the targets that researchers are interested in, so that we can actually look into that, and then we can then expand the catalog, and once the tools are available, we can let you know, and then that's where you can further use the reagent to better understand the biology or pathway of the protein that you are interested to work with. Yeah, so with that, we have a poster, a technical poster. So if you want to have a soft copy of the poster, you could scan the QR code. You could save a PDF copy of it. Yeah, so if there's any questions with regards to the Ankyrons and you are interested to learn more about it or to collaborate in the Ankyrons, we more than welcome you to reach out to us, to any one of the ProImmune members, and then we will look into how we can actually support in this area with this Ankyrons technology. Right, so with that, I will leave my contact details here, which is the same WeChat QR code that I actually left yesterday. Yeah, so feel free to contact us to let us, to explore areas that we can work together. Very much. Thank you very much.
So yeah, one thing I also want to say is that in most cases, we will generate new Ankyrons for free. Right? I mean, that's if you want a recombinant monoclonal made to a custom target, it will cost you tens of thousands of dollars. And we can generate Ankyrons to hundreds of targets each month. And if it's something that's of interest to us, then we'll simply do it. And you just need to do the clone testing at the end of it. So I think that's probably the single biggest change to research antibody technology that Ankyrons can deliver. So ultimately, we want to cover just about everything that anyone is ever interested in. And we've actually done that quite a bit for different groups in China already. So feel free to contact us, and we will review your protein targets and obviously discuss with you the feasibility of how possible that is. Integral transmembrane proteins like GPCRs are still tough because we would essentially have to generate them as targets, but we're even looking into that. So if anyone has any questions about it, then...
Yeah.
Yeah, thank you for your talk. But as far as I remember, the Ankyron repeat, there's a clinical trial ongoing. The protein is called DARPin. So is it kind of related, or is it completely different from DARPin?
Yeah, so it's a similar scaffold, and I think what we've done is we've looked at alternative scaffolds have been sort of pursued by people for more than 20 years as a way to close a gap in therapeutic development where maybe antibodies couldn't reach. And what we felt is okay, that's great. We're not a therapeutics company, But we felt that some of these platform technologies, and also ribosome display as an approach for generating binders, is very powerful for generating research tools. And there's still a gap with antibodies as research tools. And, we provide tools and services for R&D. And we felt that that's an opportunity that was overlooked effectively. And when you think about antibody therapeutics, first there were research antibodies, and then people thought, oh, okay, I can target this protein now, I can turn it into a drug. And so strangely, with alternative scaffolds, that hadn't happened. And so the idea is, well, we can close that gap. We now have a technology stack through the display, the library-based selection, direct selection from a library. So there's no, the SARS-CoV-2 data that Zubin showed, we just directly selected these clones. So we got strongly neutralizing subtype-specific Ankyrons without actually trying very hard. So that wasn't something that we put a lot of effort in. They just came out of a run where there's hundreds of other targets in it. And you get very high quality product out of it. So it's basically a technology that accelerates research. That's how we position it, and that's the sort of value that it can offer you.